Hello, I'm Janine Cody, Director of the Chromosome 18 Clinical Research Center, and I wanted to talk a little bit about whole genome analysis. These are tests that can be done to assess all the chromosomes, the whole human genome, all at once in one test. They're often done for discovery purposes when you're trying to figure out what in the world is going on. There are three types: karyotype, microarray, and sequencing, and I'll talk a little bit about each one. First, the karyotype. That's the historical way that chromosomes have always been assessed using a light microscope. However, the technology in the microscope has changed quite a bit since the early days. And what is done is the cells from a blood sample, so white blood cells, are viewed on a microscope slide. So there's hundreds of cells, and each cell creates a spread of chromosomes, kind of like this. And of course, they've been stained specially, so they show you the black and white bands. So these are the chromosomes from a single cell. And across that microscope slide are hundreds and hundreds of these chromosomes, each from a different cell. And this is a little uninterpretable. So what the computer does is they line them all up in pairs, from largest to smallest. And you can see that each chromosome has kind of a constriction somewhere along its length called a centromere. But the centromere is not exactly in the middle, so it usually creates what looks like a long arm and a short arm. And the convention is the short arm is always positioned at the top. It's also called the P arm. The long arm, called the Q arm, is always positioned at the bottom. And this particular person, as you can see in the bottom right corner of this slide, has an X and a Y. So this particular karyotype is from a male. And what you can learn from this are a couple of really important things. One is, because these are assessed from one cell at a time, you can compare many different cells. So you can determine mosaicism. For example, if you looked at 100 cells and 10 of those 100 cells had an extra chromosome, which you would see here, extra chromosome 18, let's say, but all the other 90 cells had normal chromosomes, you would find out that that person had mosaicism for trisomy 18. So this kind of test is really good for detecting mosaicism. It's also really good for detecting what's called balanced translocations, or any sort of translocation, when a big chunk of one chromosome swaps places with a piece of another chromosome, and that's often apparent here. And sometimes people with balanced translocations have no issues because they're balanced. It's an even swap, but they're at very high risk for passing on a chromosome abnormality that's unbalanced to one of their children. So it's important to know. And as I always say, if you've ever heard any of my talks, I always use this slide, because anything you can do to this picture of chromosomes with a copier, scissors, and glue can really happen, and it does really happen. So this kind of test is helpful for seeing some of those rearrangements. So again, karyotype is really important for determining mosaicism and morphology changes, or large deletions or duplications or rearrangements. But there's a problem. They have to be large duplications or rearrangements in order to see them with this technology. You can't determine exactly the position of any one gene. This doesn't point out where genes are. So in order to know that, you have to move to molecular techniques. And just to illustrate the difference, here is a very old slide. Some of you may have seen this before, illustrating the difference. We have two phone books here. These, if you remember a phone book, these are the San Antonio Yellow Pages, and I use this because it kind of looks like a chromosome. You know, there's a tab there that shows two sections, and the top section is smaller than the bottom section, and the bottom section has all these little bands. And the phone book on the left is the normal phone book. The phone book on the right has a deletion. And because of those bands, you can kind of guess that maybe that deletion is maybe at U and V, maybe. It's a guess. But what you can tell is it is at the end of the chromosome, toward the end of the phone book. Just like you can see in the chromosomes shown at the bottom that there is a deletion at the end at the bottom of the chromosome. So you get a general idea. You can tell it's not the P arm. You can tell it's not way up on the Q arm. You can tell it's toward the end of the Q arm or toward the end of the phone book. Maybe the U and V pages are missing. So another example, if you look at two karyotypes from two different people, and you know these are two different people because in the bottom right corner, the one on the left has an X and a Y, it's a male, and the one on the right has two X's, so it's a female. So we know these are two different people. And the red arrow gives you a hint as to what we need to focus on, that each of these people appears to have a deletion at the end of the long arm of chromosome 18, so they appear to have 18q minus. Just to zoom in a little bit, here are those same chromosomes. And now let's look at what we can learn from a microarray. And sometimes microarrays go by different terms or labels. Sometimes it's called CMA for chromosome microarray analysis. Sometimes they're called array CGH for microarray comparative genomic hybridization. And sometimes that's just abbreviated ACGH. And then there are SNP arrays for single nucleotide polymorphism microarrays. But what these all do, essentially the same thing, give you the same sort of information. They determine copy number. So they let you know all the places in the genome where there are 2 copies, but also where there's 1 or 3 or 4. So they're important for copy number. And the important thing to know, though, is the number of probes on the microarray. So a microarray with 400,000 probes is going to have a lot higher resolution and narrow down the exact position of a deletion or duplication. It'll have more resolution than one of 40,000 probes. So, but they determine copy number. And so what that does with regard to our phone book is like opening up the phone book and just determining what entries are missing. And in this case, it's the S's are totally missing, and the entries go from real to tree. So we know exactly which entries in our phone book are missing, and that's what molecular analysis tells us. And to show that in comparison with a karyotype, here's the karyotype we've been looking at here on the left. And to the right is a map of chromosome 18, and it shows the black and white and gray bands of the karyotype. And to the right of that is a white bar. And on the left side of the white bar are the positions, the nucleotide scale. It goes from 1 at the top to about 80 million at the bottom. And on the right side of the white bar, all those little bitty white lines you can hardly distinguish one from another, those indicate the position of each of the genes on chromosome 18, each of the 263 genes. And some of those gene names are to the right. They're not all listed there because you could never read them. You can barely read them as it is. But you can see where the position of all the genes are. So you can easily see that you can't really distinguish the position of one gene versus another on a karyotype, which is why you need to go to molecular techniques. So let's go back to those same 2 individuals and zoom in on their chromosome 18 pairs. And again, the red arrows indicate what we need to be looking at, that the bottom of one of their chromosome 18s is missing a piece. Now, if we apply the data we got from a microarray analysis to these pictures, this is what we find. So the individual on the right, on the left is the list of genes that are within the little red box there. And the gray bar indicates where they have a chromosome present, and the genes that are present are indicated in white. The missing genes, the deleted genes where they have 1 copy instead of 2, are shown in yellow. And so the individual on the left does not have a terminal deletion at the end of the long arm of chromosome 18. They have 18q minus, but they have an interstitial deletion. So they're missing those genes sort of toward the end, but not the ones at the very end. Now the individual at the right on the karyotype looks pretty much the same. But you can see that they in fact have a terminal deletion of 18q. So the genes on the shown in white are the ones they have present, and the ones in yellow are the ones that are missing. So they have a terminal deletion. And this is important because the person on the left, because they have those genes at the very end of the chromosome, they're not at risk for growth hormone deficiency or dysmyelination of the brain, just to pick a few examples. And so this has clinical impact. It informs you a little better about what you need to look out for, be concerned about. So moving on then to genomic sequencing. There's two types of genomic sequencing. One is called whole exome sequencing. And so it's all the exomes, or really, it's another word for genes, all the genes in the human genome. But what this technology does, it doesn't just measure copy number, it tells you spelling differences. But it doesn't tell you the spelling difference of the entire genome, it's only the genes. So it's like telling you the spelling, it's doing a spell check on just these circled regions, selected areas throughout the genome. So it's not all of the genome, it's what's considered the most important parts of the genome. But again, it's not the whole genome. Where whole genome sequencing is all the spelling difference across all the chromosomes, the entire human genome, and they're all over the place. As all these little red underlines indicate spelling differences from the reference. And here's the rub. Sometimes those spelling differences make a difference, and sometimes they don't. For example, gray, gray. Two ways to spell, same word, doesn't matter. Spelling difference doesn't matter. Color, color, doesn't matter. Draft, draft, same thing. Ball, ball, oops. That spelling difference, the difference in one letter, means those are two different words. So, the challenge with whole genome sequencing is going through the millions of differences in spelling to determine, do they make a difference or don't they make a difference? And that is a huge challenge when there are millions of these spelling differences for every single one of us in comparison to the reference or a database of spelling variability. So that's like doing a spell check for the entire phone book. It's a mammoth task, and that is the really big challenge of human genetics today, is determining where the spelling differences make a difference. And sometimes one spelling change may not make a big difference, but if you have 8 specific spelling changes, they may all add up to a problem. So they're not. So the analysis is really the big challenge here. And as much as we all know is close to impossible to figure out what things really cost people, or insurance companies, or whoever, these are the relative sort of general idea of the costs of these various tests, which help explain why one test might be ordered over another. Karyotypes are several $100s. A microarray is sort of an intermediate cost. Genomic sequencing, depending on whether it's whole exome, whole genome, and how much analysis gets done, it could be from $2,000, $24,000, somewhere in there. I mean, it's a huge range. So it just depends on the analysis, which, as I said, is the big challenge with genomic sequencing. So hopefully this gives you a little bit of an overview of the whole genome analyses that can be done, and what you can learn and what you can't learn from each of them, and so why your physician may order one versus another. And as always, if you have any questions, just let us know. If you email chromosome18 at uthscsa.edu, you'll get to us and we'll be happy to answer any questions. So thank you so much for your attention.